Good morning and welcome to our webinar this morning. It's our THYME Project, Sustainable Wastewater Treatment and the Bioeconomy seminar. Welcome. I'm Jenny Spear. I'm the University of Hull Programme Manager for the THYME Project. And I'd like to take this opportunity to thank Catch for their involvement in developing the programme and to Catch and Biovale for promoting through their networks. Just a little bit of background. THYME is a collaboration between the universities of Teesside, Hull and York, and it's been set up to work with stakeholders to develop the regional bioeconomy. You'll notice a few of the forthcoming talks will showcase some of the technologies that are being developed as part of the THYME project. And the challenges that the programme sets out to address are as follows. Challenge number one is transforming bio-based waste into new high-value products. Number two is converting industrial sites by repurposing them for bio-based manufacture. And the third one is growing the productivity of the regional bio-based processes. So the purpose of the webinar today is primarily knowledge exchange around the topic of sustainable wastewater treatment, and hopefully with a view to develop a network around this area if there's an appetite to do so. The talks today will focus on activity at very different technology readiness levels, and you'll see as we go through, around reducing energy and resources to process wastewater, around transforming waste from the water system into valuable products, and also improving the productivity of wastewater processes. Just a quick preview of the programme. Obviously, I'm the chair today from the University of Hull. I'll shortly be inviting Jill Mooney from Catch to give an intro into the work of Catch and different opportunities to engage with the group. And then there'll be 5 talks from across the THYME partnership. And again, you'll see as we go through, there are different technologies, different readiness levels, etc. But hopefully we'll all be engaging for you. And then right at the end, we're going to have a Q&A session. So just a little bit of information about this at the meeting today, just to let you know that it's being recorded and the link will be available shortly after. We're not going to do questions as we go. We're going to save them all to the end. But if you look on your screen, it should be very obvious there's a Q&A tab. And if you click onto that, you'll be able to pose questions throughout the talks, and we'll address them at the end in a Q&A session for the panel. There will be a short survey available to complete at the very end of the meeting. So without further ado, I'm going to come off this meeting and I'm going to make the introduction to Jill Mooney, who's going to tell us a little bit more about Catch. Hi Jill, when you're ready.
Thank you. Hopefully this technology will work. Try and share my screen. Right. Hold on 1 second. Go on. Hopefully it's going into slideshow. Okay. Hopefully you can all see that. Thank you very much. So thank you very much for inviting me to speak today and for this collaboration with the THYME Project. My name is Jill Mooney and I'm Head of Catch Technical Services. And I just wanted to take a few moments to introduce Catch for those of you who are not aware of what we do and our existence. So we were originally set up as one of the 4 chemical clusters by the regional development agencies back in the early 2000s. So there's Scotland, the North East, Yorkshire and Humber, and the North West. However, of course, all that changed in 2012. So we are now solely supported by our industries, our membership, and our associates. We are run by our board, which is industry-led, Centrica Storage being one of our members and being one of our members of the board. And you can see it's a cross-section between industry, contractors, academia, as well. We have 3 locations now. We have our main Stallingborough site, which I'm sure some of you will have visited. We have a presence at Huddersfield at the Process Manufacturing Centre there as part of Kirklees College, and also in Hull at the Aura Innovation Centre. So just a bit about the catch facility at Stallingborough. It's a variety of offering we have there, including the National Centre for Process Manufacture, which has just had a complete new upgrade. It's a fully operational processing plant and tank farm and storage area, where the apprentices who are trained on site will be trained in a fully operational area. There is also a conference centre, training centres, a DCS system and control room, and also specialist training for the industry, such as working at height, scaffolding training, you name it, we can do it basically. We also have other providers on site to aid the industry and delivery. So basically a catch is a place where industry comes together to support the industry training and technical advice, and that's some of our providers who are on site with us. With regards to catch, there are 3 main areas of catch: catch skills, catch membership, and catch technical. But we all work together for our aim to deliver a national centre for membership and technical services hub for the energy intensive industries. Our membership, we are able to offer a diverse number of networks. They are different, they are geared towards industry. You won't find sales pitches at our networks, but you will be able to network with like-minded professionals. And I'd just like to draw your attention to the 2 new networks that we have now. The Humber Industrial Decarbonisation Network that launched this year as part of the decarbonisation roadmap within the Humber region, and also launching in 2021 is a hydrogen network. So we have over 50 plus client members. We host 60 plus events and networks per year. And of course, since COVID, we have had to go virtual as we are today. And we're now offering a blended version as well, both online and face-to-face. And over the past year, we have supported 600 plus people and organisations. Catch Technical, you may not have heard of, and this is a department I head up. That was launched last year, and it's a carefully selected team of specialists providing support to the businesses. Again, this was as a direct request from our members. We have consultants from across sectors and cross industry, and we were able to offer consultancy from hazard and HAZOP studies, human factors, lean implementation, asset management, safety and health, including mental health, reliability, maintenance, you name it. We can help you with a project that you need to implement in your business. I have now 17 plus consultants on the consultants bench. We have now over 300 plus years of experience, and we are able to offer you one solution to your business if you wish to implement a new project or a new initiative. And then finally, I just wanted to touch on CONCOM. So if you're an operational site or business, you obviously have contractors who come onto your site, and you need to ensure that they are competent in the work that they will be delivering for you. And that is what our CONCOM scheme does. It's a single standard audit that we can audit the contractors through. We currently have over 240 plus contractors on that scheme, and we manage it on behalf of our 20 plus client members, and we deliver it and have been delivering it now for nearly 15 years for them. So it's a well-established contractor competency scheme. thank you for your time today. I didn't want to waste any more time. I wanted to get straight on to the bioeconomy and wastewater treatment. But if you've got any queries or any questions, please get in touch with me. And I'll hand over back now to Jenny.
Thanks, Jill. That was a really excellent and succinct introduction to the work of Catch. And I think I can see lots of opportunities for us and partners to get involved in and really take advantage of your fantastic networks. So thanks for that.
Thank you. Yeah, it's all about collaboration.
No, absolutely. And talking about collaboration, that segues almost perfectly to Jai Waduan and student Reese Ward and industry partner Adam Beardmore from Centrica Storage. So Jay is from the University of Hull, and he's going to specifically talk about the novel process that they're developing in partnership with Centrica.
Thank you so much, Jenny. So let me just share my screen. Perfect. Hopefully you can all see the slides. Yeah. Well, good morning. Our work at the University of Hull in industrial wastewater treatment has been undertaken in collaboration with Centrica Storage Ltd through a very particular type of Innovate UK scheme. Here today are, as Jenny mentioned, Adam Beardmore from Centrica Storage and Reese Ward, who works at both Centrica Storage Ltd and the University of Hull. If you'd like to say hi, so you know who you are.
Morning, everyone.
Good morning.
So we have been undertaking this collaboration through a knowledge transfer partnership, a KTP, a way of bridging the gap between university discovery and real life applications or commercialization of that work in an industrial setting, potentially improving productivity, profitability through the creation of new products, new services, new technologies, whilst capitalizing on the resources and skills available at a university. Our project on wastewater treatment actually marries up neatly with wastewater problems in the bioeconomy. If we consider the oxygen demand and actually consider it on a transient basis, so if you take the chemical oxygen demand and multiply it by the flow rate, we realize actually that the oil and gas industry is comparable in terms of its transient oxygen demand as a large variety of the bioeconomy itself. So that any innovations made in any one of those industries for wastewater treatment can actually be transferable between the two differing sectors. So I'm going to hand you over to Adam, who's going to talk to you about Centrica Storage Ltd and potential issues that they face with their produced gases.
Yeah, thank you, Jay. So good morning, everyone. Just to give you a bit of background on Centrica Storage, if you haven't heard of us. So we are an oil and gas operator working with our head offices in Hull. What you can see in the pictures here is we have a operating Easington gas terminal based at the coast, and that brings in gas from the southern North Sea. So we have a number of fields that feed into that terminal. The main field you can see on the left there is our operated asset, the Rough field, and that's where this kind of project looks like. So that's a free jacket platform, 24 wells down into a subsea reservoir. Previously used for storing gas, but currently we are just producing it. And both of these assets have to deal with wastewater. Both of them have challenges in processing that. So I think if we go to the next slide, there's a bit more, yeah. So the offshore assets built 25 years ago, kind of old historic tech, most of it quite basic, quite simple. The produced water comes from the gas streams, so they're quite hot underground. As you bring them up, they cool, you get dropout of condensed water. These then go through a number of knockout vessels, and then finally the last stage is a filter. this filter is there basically to take any sort of hydrocarbons or benzenes or nasties down to around 30 parts per million before the water is put back into the sea. It's a very kind of challenging operation to change these filters out. As you can imagine, they get compacted with high concentrations of kind of nasty hydrocarbons, so breaking those to remove them, it requires full kind of PPE and hazmat sort of kind of controls for personnel. It's classed as hazardous waste, so when removed, taken back on shore, it has to go for processing as well. So quite intensive in terms of the risk and the amount of effort to just filter the water itself.
Okay, so we engaged with Centrica Storage Ltd, and they explained their problem to us. They invited us to their facilities so that we can actually realise the scale and magnitude and actually identify those issues on site as well. We proposed a plan of work in which we could actually use some of the university's experience and expertise, particularly along the electrification of technology, to overcome some of the issues that Adam has mentioned, and develop this as part of a knowledge transfer programme, a UK government, a part UK government sponsored initiative that encourages this collaboration with industry to be developed. The KTP is a four-way partnership between the industry partner and the university that the government helps to fund. In the case of a large organisation, 50% of the funding comes from government. In the case of a small medium enterprise, something like 67 to 75% of the funding comes from government itself. And the purpose of this project is to cement the collaboration through a transferable person who is known as the KTP associate and happens to be a recent graduate. This graduate is kind of supervised by both sides so that both industry and academia can converge together on providing a real and tangible solution to the industrial problem. KTPs are audited every quarter, more or less, so as to ensure that the project is on track and realising the expected benefits to the company, to the university, to the associate, and crucially, as a result of all of those benefits being accrued, the government as well. For our particular project, Rhys was appointed to be our KTP associate, so I shall hand you over to Rhys for a few words.
Yep, so I was appointed to be the KTP associate here after I completed my degree in chemical engineering from the University of Hull. And working on such exciting and technically innovative projects is hugely rewarding, both with the company and with the university. And on top of this, as the associate, I actually have access to bespoke advice from a government mentor, government advisor, and have also received a personal development budget as well to enable my further upskilling.
So we're currently halfway through our project with Centrica Storage Ltd. Our innovations have really derived from our own backgrounds in process electrification, so that you can use electrochemical routes to treat the water. And we're actually extremely efficient. Lab tests suggest that we can get rid of in excess of 90% of the nasties, so that we're actually better than or comparable to existing technologies. Our innovations over the 3-year project that we had put forward are actually currently being scaled up. And we're doing that so as to do two things. First, increase the technology readiness level, and second, ensure that we have the capability of deploying it in the highly regulated ATEX compliant facilities that Centrica own in both of their assets. And we're doing this not on the university campus, but rather at the university's Aura Innovation Centre, where Catch also have facilities. This low carbon centre allows us to have workshop facilities to do the research and testing that we need to actually develop and deliver on this particular project. So it brings the skills, knowledge and expertise needed, and it's a real benefit that it's located just a stone's throw, would you say, Adam, from your courses at Woodland House,
Yeah.
on the same campus. So thank you so much. Just want to say that if any of you are interested in applying for a KTP, we have an amount of expertise at the university. You can feel free to get in touch with Yosina or Tracy, or even me. We'll put you in touch with the relevant person. Thank you. I'll pass you over to Jenny.
Hi there. Thank you very much for that. I was just reflecting actually that the power of this sort of, it's not just funding, but this targeted support and the way in which it can unlock these collaborations and to answer real world problems. I think it's really exciting. And I must apologise to Reese because I referred to you as a student, and I should know better because I used to be a KTP associate myself many years ago. So no, but that's great. It's a fantastic opportunity, and I'm sure for those of us coming out of fundamental science and some you know, translational research, that actually looking to embed our problem solving and our relationships with industry, that KTPs are a great way to take that forward. So thanks to everybody involved in coming to talk about that. Before I carry on talking about KTPs, of which I have a great passion for, I think the next talk is going to be University of Hull academic Pedro Beltran Alvarez. He's going to talk about a small time research project that he's been carrying out in partnership with Teesside University, and it's a much more scientific project around understanding molecular codes of microorganisms and how we can use this information to boost the productivity and efficiency of microorganisms in wastewater treatment. So without further ado, I shall pass you over to Pedro.
Thank you very much, Jenny, and thank you to Tilly, Amy, and all the team for organizing this. I'm absolutely delighted to be able to share with you this time on our work on the PTMs of bacterial in the wastewater treatment setting. So yes, I'm Pedro, I'm in biomedical sciences at the University of Hull. And as I said, I'm the group leader of this PTM research group. PTMs stands for protein post-translational modifications. And the analogy that I always use to explain PTMs is that they are like the clothing of proteins. So when proteins are born, when they are synthesized in the cells, proteins are born without any clothing on, naked, just like us. But of course, before they go to work, before they go and socialize with other proteins, just like us, we put some clothing on, they put some clothing on, they put some modifications on. And these modifications are normally reversible and dynamic. So if it's very cold, we put a jumper on. Proteins can do the same. If it's very hot, we can take that off. Again, proteins can do the same. And that's a way that proteins adapt to the environment. So the hypothesis, which is underlying this project, is that bacterial proteins change their PTMs, change their modifications to adapt to the wastewater treatment environment. And this is based on some preliminary data that we published last year as preprint. The opportunity that we identified was to target that third time grand challenge that Jenny has mentioned before. And in particular, a research question is, can bacterial PTMs be useful biomarkers of wastewater treatment efficiency? We haven't done any wet lab analysis. We haven't collected wet samples. We have used an alternative approach, which is known as proteomic data mining. So we have used proteomic data publicly available in PRIDE, in this proteins identification database. And that is a database which captures all proteomics data that researchers produce, all those raw files, all those raw data, or not all, but if it's a decent study, they are deposited in these public repositories. And then we can go with a different research question and interrogate, investigate those raw data to try and answer the question that we are interested in. And again, I've got an analogy to try and explain a methodology if you're interested, but as I said, I'm not going to explain that here because I only have 7 minutes left. But if you're interested, I can expand on that. And we are using proteomics software, including MaxQuant and Perseus, which are again free and available to everyone. In terms of results, we have identified several projects available in PRIDE, in that proteomic database, which have investigated bacterial proteins in a variety of settings, and as you've got on the screen, so including wastewater treatment plants, and also the human gut, so essentially fecal samples, biogas plants, and then soil and compost. And we have used proteomics software, MaxQuant and Perseus, to identify the bacterial PTMs, the modifications, the clothing of those proteins in the different settings. Now, I have to admit that we were a bit ambitious when we set up this project, and we wanted to do quantitative analysis. So we wanted to measure the levels of those PTMs in bacterial proteins in all these different environments and settings. Simulations are, this proteomic mining is very computationally demanding, in fact, more than we expected. So we were running simulations for 6 and 7 weeks, and then they would crash, which was very, very painful at that time. So we went to our contingency plan, and that has meant doing qualitative comparisons only. And by qualitative, the way that we have worked around that is we have identified and we have quantified unique modifications in all those different environments. So unique PTMs to bacterial or to bacterial proteins in wastewater treatment plants and gut and biogas and so on. This is a summary of our results. So what you've got here is the different settings that we have identified or investigated: wastewater treatment plants and biogas plants and human gut, compost and soil, and 4 different PTMs, 4 different types of protein clothing. So we've got PTMs affecting different residues in proteins, including serine, threonine, tyrosine, and arginine. What you can see, I hope, is that the highest number of modifications, the highest number of unique PTMs is consistently in those bacteria, in those bacterial proteins coming from biogas plant samples. So, for example, there are 35 unique methylation events in those bacterial proteins. There are about half the number of unique PTMs in the human gut. And in this case, there are essentially no unique PTMs in WWTP, compost, and soil. And it is a consistent picture across the different PTMs that we have investigated. So what I've done here is I've included the ratio, which is the number of unique PTMs in the different settings, and normalized to the biogas plants, because that's the highest number of PTMs, of unique PTMs. So overall, we can see here that as I said, the biogas plants can support the highest diversity of unique PTMs, so that's why it is 100. There are about half those numbers of unique PTMs in the human gut, very, very little really in wastewater treatment plants, compost and soil. As I say, that's across all the PTMs that we have identified. What does that mean? We need to remember that clothing is expensive, so PTMs are expensive to the cell. And just to give you an example, it takes 12 molecules of ATP for any methylation event. ATP is the coin, yeah, the money in those cells, the energy in those cells. So PTMs are expensive. And in our study, we have found that conditions in the biogas plants are the best where bacteria can thrive. And we can see that because that's the, we could identify the highest number of unique PTMs. And that's what I've tried to represent there in those circles, yeah, which are proportional to the number of unique PTMs in the different settings. So biogas plants are the best conditions. Wastewater treatment plant, and this particular wastewater treatment plant that we have investigated, which was one in Germany, my hypothesis would be that the biological process in that wastewater treatment plant was not very effective, because, as you can see, the number of unique PTMs was very, very low and essentially comparable to compost and soil. So our overarching question is, can PTMs be biomarkers of wastewater treatment efficiency? That question was posed by some authors last year. They were proposing to use proteins as biomarker. We are going a step forward and proposing to use PTMs. So I hope I've convinced you that we have developed, or we are developing an original idea to measure wastewater treatment efficiency based on those PTMs. It is good value for money in the sense that we had a small grant, only £2,000, and we have generated gigas of data using proteomic data mining. And we want to develop that into NERC and BBSRC grant applications to understand if these PTMs can be the biomarkers of those wastewater treatment plants that we are looking for. So I'm going to stop there. Thank you very much for being there. It's been an absolute pleasure to be able to have this opportunity, and thank you to THYME for funding this PTM research bridge between Hull, Teesside and York. Thank you, everyone.
Thanks for that, Pedro. And it's fantastic to have those results hot off the press. I can see there's a huge amount of further analysis to do. So it's a pleasure that THYME can kind of help act as a catalyst to get the initial work going, and let's hope it really sort of leads to some substantial follow-on funding to carry on that collaboration. Thanks a lot there. Great. We now move to James Chong from the University of York, and again he's going to talk about a number of collaborative projects that he's been involved with through THYME, and it's really going to focus on how to better define the mechanisms of anaerobic digestion in wastewater treatment, and hopefully I think we'll allude to some of the ways in which we can improve process yields within that system. So thanks for that, James.
Thanks, Jenny. Morning, everyone. I will try and share my screen. Okay, great. So as Jenny said, I'm going to cover a couple of projects that THYME has supported that I've been involved with. Oh dear. Okay, so this is the process that we're interested in, the production of biogas from organic waste. That's a microbially based process. Basically takes long molecules, they're broken down by different parts of the microbial community into smaller molecules, which ultimately result in the generation of biogas, which is a mixture of methane and carbon dioxide. Generally, just over half of it's methane and most of the balance is carbon dioxide. And there's some interesting unique biochemistry that goes on by a group of microorganisms called methanogens, which can use a variety of interesting pathways to make methane. Okay, and we can harness that process, and we do harness that process in domestic wastewater treatment. So here you can see an overview of Yorkshire Water's Nostrop site, and in the background, these 4 white tanks are used for anaerobic digestion at a scale of 7.5 million litres per tank to produce biogas, which is then collected into this bubble and used for running a combined heat and power engine, so both producing electricity and heat. Okay, so for those of you that are not aficionados of domestic wastewater treatment, the way this works is the water comes onto site, it goes into a settlement tank where the primary solids are allowed to settle out. That dirty water is then decanted into a second tank, which is aerated. That also basically removes a lot of the organics in using an aerobic microbiology process, and those microbes form biomass which can also settle out, and they form what's called a secondary sludge. Just as an aside, the air that we blow into those systems uses 1% of all the electricity that's consumed in the UK, so this is a fairly energy intensive process. The clean water from this process is decanted again into a trickle bed filter, more microbes involved at this point to really polish that water and get rid of any straggling organic molecules that can be removed. And then that water that remains, the final effluent, is decanted out into normally the river discharge. And that water is cleaner than the water that is abstracted upstream to make drinking water in the first process. So both the primary sludge and secondary activated sludge are collected, and they're put into an anaerobic digester, which is the main interest in my group. This also contains a microbial community that produces biogas and a digestate, which is nitrogen rich and can be used potentially as a fertilizer. Okay, so in order to understand this biology, we have collected a number of samples from around the Yorkshire region going to Yorkshire Water sites. We've used DNA sequencing rather than looking at proteins, as Pedro was talking about, or PTMs. And based on DNA sequencing, we can see that there's a large variation in the number of organisms that are found within anaerobic digesters. The population within the digesters is different to the population that's found in the material going into the digesters, and the digesters themselves vary depending on how the material is treated before it goes into the digester. So the first project that we were involved with that has had THYME support is led by Caroline Orr at the University of Teesside, involves her collaborator Helen Carney. It also involved the Biorenewables Development Centre, which is based in York, and industrial partners, Northumbrian Water, who were interested in whether we could use these molecular techniques to improve the efficiency of their AD processes. So basically, the proposal was Northumbrian Water run 2 anaerobic digestion sites, one at Brand Sands and one at Howden. Their material is heat treated before it goes into the anaerobic digesters, and the reason for doing that is to try and break down particularly the microbes in the secondary sludge, which tend to be rather difficult to access for anaerobic digestion. So Caroline proposed that Northumbrian take samples of the material coming into the sites, the material after it was heat treated by the Canby process, and then from the 3 digesters that were receiving that material. And those samples were taken once a month over 5 months in the summer of 2019 from both sites. And the reason for looking at both sites is because they perform differently, even though, you know, theoretically they're running exactly the same process. York's role in this was to measure a number of the parameters from these samples that were collected. We were looking at chemical oxygen demand, carbon and nitrogen totals, and the volatile fatty acids, so the small molecules that are produced as part of the microbial breakdown into biogas. At Teesside, Caroline arranged for the extraction of DNA from all these samples. That DNA was provided to the BDC, who have a 16S RNA sequencing service, and then all the data are gradually going back to Teesside. And at the same time, we've started looking at some other technologies that we've been developing in York to try and understand the AD process. Okay, so the samples were all collected very successfully and distributed, and then before they were completely analysed, coronavirus hit. So we're not at the end of this project quite yet. We're still analysing some of the data, but I can give you a brief taster of the kind of things that we've managed to collect so far. So here we're looking at the volatile fatty acid analysis of all the samples. You can see basically a time course at each of these stages across the two sites that Northumbrian operate, and the different volatile fatty acids, so acetic being the smallest and heptanoic being the largest, and how those concentrations change over the course of the sampling of 5 months, and also how they change through the process of digestion. And as you would expect from the way that the process works, these volatile molecules are gradually broken down in the digester and converted into biogas. But what you can see is actually a lot of these volatile molecules are lost during the Canby process, or at least converted into other material that are not volatile fatty acids that are being fed into digesters. And you can hopefully also see that there's quite an amount of variation in how the digesters are operating, particularly at Howden, which suggests perhaps that there's a difference in that mechanical process. Perhaps one of the digesters is being fed at a slightly different rate to the other two digesters. Right, as I said, this is still a work in process. One of the other technologies that we're looking at this is an assay that we developed for another project. This is a LAMP assay, so very rapid, potentially colorimetric assay that tells us about the specific presence and absence of particular species within any sample, but in our case, within anaerobic digesters. We had developed this to follow specifically the methane-producing organisms in another project that we ran with the University of Southampton, which is to do with biogas upgrading. I can't go into details here, but basically the idea is that you can take renewable electricity, use that to make hydrogen, which is very difficult to store, but then feed the hydrogen into digesters, and the microbes will adjust to that and combine the carbon dioxide and hydrogen that you're injecting to make methane. So instead of getting the typical 55 to 60% methane, you end up with 95 or better % methane coming out of the system. Right, so doing those projects highlighted a challenge to us, which is we do most of our experiments in the lab, and this is the facilities at the University of York, which have been sponsored by Yorkshire Water. We have 60 5-litre digesters where we can do experiments. Yorkshire Water also have a pilot rig, which allows us to scale up from 5 litres to 1,000 litres. But obviously there's still an enormous step getting up to process scale, which, you know, in Yorkshire Water's largest installations is 7.5 million litres. So the second project that we have been involved with, sponsored by THYME, is to look at this idea of repurposing process scale assets to demonstrate AD innovations. This was done in collaboration with Peter Ball, who's a colleague in the management school at the University of York, Gary Montague at Teesside, Tom Taylor at Yorkshire Water, and actually most of the work was done by Nathan Innard, who worked for me at the University of York. So the idea here is that as Yorkshire Water is updating its sites and rationalising processes, it's going to close some of its smaller AD sites. Normally those facilities would be decommissioned, and the question was whether any of those facilities would be suitable for using for demonstrating innovation, which would allow us to bridge the gap between pilot and process scale without any of the risk of just asking someone to make a change that looks like it works at small scale into a process plant, which obviously, you know, nobody wants to be responsible for losing sewage treatment facilities, for example, at Sheffield or somewhere else. It would not make us very popular. Okay, so Nathan conducted a survey of potential assets that were being closed down. We applied a number of different criteria to work out which sites might be suitable based on the current state of repair of some of those assets, whether or not there were 2 or more digester tanks available so that we could do controlled experiments and actually demonstrate that our innovations were improving the process. And we basically narrowed it down to 3 potential sites, and then did some further work which allowed us to narrow it down to Bridlington. So here you can see the Bridlington site. There are 2 anaerobic digesters, which are about just over 1 million litres each, which is, again, a nice step up from 1,000 litres and brings us into the realms of process scale. And you can see there's plenty of room on this site potentially for redevelopment and for introducing plug and play type facilities so that other innovators could bring their equipment on site and we could introduce it into that AD process. We're in the very last stages of generating a report for this work that we've done through the THYME project. And the next step is obviously to source funds to try and actually realise some of these ambitions. We think we're going to be able to do this. 2 things have come out fairly recently. So Ofwat, the water regulator in the UK, has just established this £200 million innovation fund to try and encourage innovation within wastewater services within the UK. And at the same time, all the water companies in the UK, which traditionally have, I guess, had a little bit of healthy competition and not wanted to therefore share necessarily the results of their innovation, have drafted this idea of innovating together, and core to that they have proposed a UK National Water Centre of Excellence, which they propose would be a virtual centre that could make use of facilities all across the UK for the kind of work that would allow people to innovate. Okay, so that's all I have to say. And I'll pass you back to Jenny. Thank you very much.
Oh, thanks, James. We always think of anaerobic digestion as being a really kind of stalwart process, you know, that's been around for many years, and I think it's great to hear that there's still so much room for innovation within that process. And I think, thank you for sharing the information about the process scale facility under development. I can see room for plenty of collaboration around that facility, so I'm sure you'll be inundated with queries there. And again, thank you for posting about the funding opportunity, because it always greases the wheels. So thank you. And I'm sure there'll be plenty of questions at the end of the session for you. Okay, our next talk is back to the University of Hull. It's Andrew Bower, and he's going to talk not necessarily about THYME Project, but about an EU-funded project, solid sediments, and a novel use of spores to remove pollutants such as pharmaceuticals from the water system. So, Andrew, it's over to you.
Okay, right. Thank you very much then for the introduction. As Jenny was saying, I've been working on a project for the last about 3 years, which was funded by the EU. And what I'm going to tell you about is actually only a very small part of it. But the general theme of this project, Solid Sediments, was about water treatment gone wrong in some ways, in that very small amounts of pollutants do still end up in the waterways and can cause issues if they are endocrine disruptors with the microfauna, and ultimately if it gets into the oceans to fish as well. And so I was coming into this project through an interest of the materials which come from plant spore or plant pollens, and these are could be crop products or byproducts from resources And so we've worked in a lot of different areas. And in this case, I'm going to tell you a bit about how we can use them for water treatment. And probably the best way is by giving an introduction to the project through a video. So that gave us a little brief introduction to the project. We mainly work with lycopodium powder, which comes from club moss. It's commercially available. Of course, it's a crop, so the amount that you can buy depends on how much of it actually is grown, but it's actually used in pyrotechnics as well as a dusting powder, and we buy it in multi-kilo quantities already from a range of commercial suppliers. Now, pollens and spores are of interest because they make up quite a lot of the bulk of organic matter which is found in the sediments in riverbeds, and materials are adsorbed onto this organic matter and then reside for quite a long time in the sediments. So we thought that we might be able to use this as a way of being able to basically have a fine way of removing materials in a cost-effective way from wastewater. So as was shown in the video there, these particles are quite complex structures, and what we need to do to make them hypoallergenic is to remove some of the materials on the inside and the outside. And we have a range of different ways in which we can process this material to end up with the empty capsules. This is a very old classic extraction, but we have much more efficient, much quicker ways now of processing these materials. They have narrow channels through them, so this is a cross section of one of the walls, those microcapsules, so material is able to be adsorbed and absorbed very readily. And also they have a range of chemical functional groups on the surface. And these mean that these particles are very amenable to a range of different modifications to tune them to adsorb different types of material. So in this particular project, we have a range of materials. So you can see they're slightly different colors depending on what type of treatments that they've been through. But in some cases, they are more lipophilic. Other cases, they're more hydrophilic. And they have different abilities to adsorb materials which themselves might have different properties. Now, one of the earliest pieces of work we did before even the solid sediments work is illustrated in what happens when you take these particles and mix them with a commercial food oil. In this case, it's cod liver oil. And these particles are able to soak up the oils really, really efficiently. So here we've got some of the powder which has been mixed with oils ranging from a 1-to-1 weight ratio all the way through to a 6-to-1 weight ratio. And that's the 6 is the oil and the 1 is the particles. And you can see that 1-to-1, 1-to-2, 1-to-3, we still have a powdery type material. It gets a bit sticky at 4-to-1. So these particles are absorbing 4 times their weight in the hydrophobic oil before they then start becoming pastes and sort of like oily mixtures. So they are able to encapsulate a lot of this material. And in fact, if you take an emulsion, an oil in water emulsion, and add a few of these particles and mix it up, you'll find that that emulsion is broken and the particles will absorb those hydrophobic materials very readily. Now, we were interested in, for our solid sediments program, which is funded by the EU, on the so-called watch list chemicals. And one I'm going to illustrate here that we looked at was triclosan, an antiseptic which is found in lots of creams, toothpaste and deodorants, although it's used less and less now. So what we did was we've done some very early lab-based studies where we were comparing what happens if we take a solution of the material with some of our particles, just mix them and allow it to reach equilibrium. So we can measure what is stuck to the particles and then what is remaining in solution. And so with the case of raw pollen, you see that with the amounts that we've carefully calculated so that we can observe what's going on here, that after a few minutes, we basically see an equilibrium being reached. And in this case here, about just over 40% was adsorbed to the raw pollen. and 60 or 55% or so was remaining in solution. But when we took our processed particles, where we have removed the inside, so we've got our exine capsules as we call them, the efficiency was much greater, so they were able to absorb much more at the same rate. And of course, this is a batch type process, so we wanted to think about what's happening when we do a filtration type process. So in this case, we made a packed bed and we're passing our solutions through the packed bed. And what we can see is we've got two different flow rates. It's the same experiment, same concentration. I should say 10 milligrams per litre is vastly in excess of the levels that you'd actually see in polluted water, but it just makes our analysis a lot easier. You can see that the bottom left-hand side here of the graph, when we're passing material through, we're basically not seeing any of the triclosan being eluted, telling us that it's being adsorbed to our particles. And then the breakthrough occurs after about 175 millilitres, 160 millilitres, where we then slowly see the concentration increasing. But even after passing through, quite a large amount of solvent, this was only, with a packed bed of maybe about sort of like a quarter of a centimetre. So, actually a very sort of small amount of specks. We never got to the point where we actually had material or concentration of the solution coming off was the same as what we put in. And we were able to then calculate basically a capacity of our material to adsorb the triclosan. We were also able to take the material and just simply clean it with an aqueous ethanol wash to remove any adsorbed triclosan and repeat it. So we did this 3 times, and effectively the efficiency is the same from the first and the second and the third go. And you can see obviously within errors here that the capacity of the recycled material to the triclosan was about the same. How does it work? Well, obviously we can postulate about the type of interactions which are for any particular individual molecule, which might make them be adsorbed onto the particles. So there'll be hydrophobic and hydrophilic type interactions. But the main thing here is we can engineer these particles to try to adsorb materials of different polarities. Just as a rough comparison, I don't want to make too much of this because of course the experiments were done very differently, but comparing with granulated activated charcoal and biochar, the adsorption capacity for our material was a lot greater. Of course, there's differences in surface area and exact experiments. So again, don't make too big a comparison, but I would say that they can at least compete with those materials. So that's just a little bit there telling us about the work of the Solid Sediment Project. There are in fact actually a range of other things we've looked at, different types of spore or pollen capsules. We've looked at different types of materials. Some are better, some are worse. We've done some work which is not yet complete with Yorkshire water and Northumbrian water about what happens when we use real life water coming from the wastewater treatment plant, and also what happens when these particles go through anaerobic digester. Very interesting to hear James's talk earlier. So basically I'd like to thank Amelia who did all the work here, Jeanette Rochel is the project lead for solid sediments, and then Graham Mackenzie is a colleague in the chemistry department here who pioneered a lot of the work that we've done with the sporopollen in exine capsules. And if anybody's interested in more about our project, get your phone out quickly and scan your QR code there. That takes you to the dedicated website, which gives you a lot more about the whole of the program, as well as about this particular sub-work package here on the spore exines. And I think I've run over by a couple of minutes. I ought to stop sharing, but if you've got any questions, please do ask them through the forum.
Thanks for that, Andrew. It looks like it has great potential for this particular sector in cleansing wastewater. So I'm sure there'll be plenty of questions in the forum, but I think at this stage we'll move on to Pip Betts, who is going to be talking about sustainable drainage solutions and her role within the Flood Innovation Centre at the University of Hull. So thank you, Pip.
Thanks, Jenny. I'll just share my screen. Bear with. Right, I'm hoping you can see that because I've lost your faces off the screen. Hi, my name's Pip Betts, and I manage the Flood Innovation Centre, as Jenny said, which is part of the Energy and Environment Institute at the University of Hull. A big thank you from me also to Jenny and Tilly for the invite to take part today, and also to all of the previous speakers for some really fascinating presentations this morning. So I'm going to talk today about water treatment in the context of flood resilience, which I'll sort of talk a little bit more about the linkage there in a second, but that will lead into the support that our project, the Flood Innovation Centre, can provide in terms of developing innovative flood and wastewater solutions. So in thinking about preparing a presentation today, I was chatting with colleagues, and one of my colleagues, Kate, came up with the notion that she'd picked up from Mary Douglas in Mary's book, Purity and Danger, written in the 60s. I think Mary's widely recognised as, or credited as coining the term that waste is matter out of place. So you can see there's a picture, it's a dining table, there's a pair of shoes on the dining table. On the end of somebody's feet, those shoes are very much in context. On a dining table, they're very much not. And that follows directly into how we think about flood. So flood is very much water out of place. You've got a photograph here of the King's Arms, not picking on York, I'll come back to York a bit later today, but you've got the King's Arms there on the riverside in York. You can't see, but the ooze in this first picture is very much in its place. The built environment has been created around the river. There's the nice walkway, the seats in place. You can enjoy a drink looking out on the waterside. And then obviously the second picture, we've had a flood event. The ooze has burst its banks, and that water is very much out of place. So flooding in that context is water out of place, it's wastewater. Pretty much a regular event, I'd say, at this pub here and those other businesses that are affected along the side of the river there. So they will be, well, obviously it'll still be a catastrophic occurrence for them, but they'll be well used to that happening. But we are seeing, as a result of climate change and rising sea levels, this is happening to more and more of our businesses, our homes and our properties. And this is where the Flood Innovation Centre was born. So located in the EEI, the Energy Environment Institute, along with Jenny and the THYME Project. Our work is similar to what Jay described earlier, the KTP support that the university provides, but we're very much a kind of mini KTP, if you like, and we can do kind of a KTP light. So it's all about joining up the work and the academic expertise from the Institute and other faculties across the university to collaborate with businesses to explore new solutions to improve our resilience to flooding. We've got some dedicated facilities, but also can access expertise and lab space across the campus, but also like Catch, and like Jay earlier, he flashed up a slide, we've also got a facility at the Aura Innovation Centre just by the Humber Bridge there. So a good hotspot of everything that's going on in this area there at AIC. So thinking a little bit more about the theme of today's webinar and sustainable wastewater treatment, there's a real opportunity here for crossover between this and sustainable drainage and nature-based natural flood management solutions. So these simple diagrams I've got here illustrate in the first picture the natural water cycle, and in the second one the urban water cycle. I'm sure it's something you're all familiar with from kind of school, flipping backwards and forwards, school geography there. As we've removed our natural spaces and increased the amount of hard and impermeable surfaces through urbanisation, we've decreased the opportunity for infiltration. So as it rains, that the water is no longer able to infiltrate as good as it used to be able to into our surfaces. So, and as we've diverted rivers from their natural courses and removed the natural bends, so you can see in this sort of second picture, the rivers are straighter, so water velocity increases, and we've got that. It means much greater destructive potential for when rivers are exposed to heavy rainwater and they burst their banks, and the water moves much faster. So removing wetlands and tree cover has also affected this velocity and how much runoff, how much and how quickly the runoff ends up in our watercourses. And as the demand for housing increases, we get that places increasing demand on land. And so more and more developers are looking at and are given permission to build on floodplains. So we have some real issues with properties being at risk from flood right at the start of them being developed. So we need a system that moves us back from this urban water cycle back towards this more natural water cycle. And that's where SuDS and natural flood management comes in. So what are SuDS? SuDS originally stood for Sustainable Urban Drainage, but the urban has been kind of dropped, but it's been retained for the purpose of the acronym so that it's easier to say. But essentially, it's an approach to managing rainfall that replicates natural drainage back to those pictures that we just looked at, away from that urban water cycle and more back towards a natural cycle. So quite a busy slide here. I think we're circulating the slides and recording the presentation today, so you can revisit these at your leisure. But SuDS and sustainable drainage offer a lot of advantages over more traditional drainage solutions. As you'll see from the diagram there, the 4 pillars of SuDS are all about managing water quantity, water quality, and in turn how you can make improvements to the public realm and the amenity of a scheme, and also enhancing the biodiversity through planting schemes and kind of encouraging wildlife in those spaces. So when it comes to sustainable wastewater treatment, SuDS can play a key role, particularly in relation to water quantity. So that's slowing the flow of the wastewater to reduce flooding and the speed that it re-enters the watercourse, but also water quality. And both of these, as I say, can lead to improved environs and biodiversity. So picking up particularly there on the water quality management benefits, and I think from picking up on some of the presentations we've heard today, in common with how we've interrupted the natural water cycle, building our urban environments, so too our activities, as we've very much seen from the previous presentation, these lead to numerous pollutants entering the water cycle. This wouldn't traditionally be managed, it wouldn't be managed by the traditional pipe drainage system, and those pollutants or contaminants can wash down into the sewers and into the watercourses, and as we've seen, end up in sediment in the riverbeds, and obviously the water quality suffers as a result. SuDS can provide a means of bioretention, so removing these pollutants and managing and treating that runoff. So I've got a little example of that. Again, not picking on York, but when I was Googling for pictures, this was one that came up. So this is the bus station at Rawcliffe Bar Park and Ride. And this was a case study that features on the SUSDrain, which is the Sustainable Drainage website, SUSDrain website, about a SuDS solution that was put in place there. So there's a filter strip, a retention pond, and a reed bed in place at that Park and Ride bus station that treats the runoff that comes from the car park, so the kind of pollutants that would enter into the watercourse from the car park, and the effluent is then reused. The effluent's treated by the septic tank and then reused in the toilets and the washrooms at that bus station, and then what is discharged into the watercourse meets European standards of clean water. So an example there of how SuDS can be used to treat wastewater. Another area that we're working on, and this is with Jenny's project THYME, is looking at how soil resilience can play a role in water treatment and flood resilience. So this is a project that THYME are working on. We're having some involvement with that improving soil resilience to climate change. I won't go into this in great detail, but from a very quick slide here, essentially this is, and I've seen on Twitter that Josh is out taking pictures and taking soil samples this morning, but essentially in the first diagram here, you can see a very impacted soil structure, and the project that we're working on is looking at different types of crop selection that can have a different effect on the soil structure. So we're moving away from that kind of non-porous, very compacted structure that you see there towards this more porous and permeable soil structure, which obviously has some great opportunities for water retention and flood management through planting schemes. So a couple of examples there of the types of products that we could work on, a little bit of a whistle-stop fly-through of how the Flood Innovation Centre might be able to help in this space. My contact details are on there. We definitely need a QR code so that you can point your phone at the screen. But if you've got any ideas or innovations, or you just want to talk to us in this space, we'd love to hear from you. Thanks. I'll stop sharing my screen.
Thanks for that, Pip. I think it's great to see the diversity of all the different projects and scale at which we're working. And I can see it really ties in with businesses and planning and building infrastructure that are actually quite a simplistic solution, how you can help with that, with designing technologies to support that. So that's fantastic. Thank you very much for that. We're now at the end of our formal talks, and it's really a chance to pose some questions to our panel. I've personally got lots of questions, but let's go with our participants first. And we do have a question on the Q&A tab right now. Pedro, are you able to see that question that's posed to you from Anna Alessi?
Yes, thank you, Anna, for the question. So the answer is yes, we have generated gigabytes of data, including the identity of those proteins which are modified and the site of the modification, so the specific amino acid which is modified in those proteins. We are analyzing at the moment the type of proteins that they are, so whether they are, say, nuclear proteins or cytosolic proteins or involved in a particular process within cell metabolism. And as I say, the general idea is that the higher the number of PTMs in those bacteria, then the more efficient that biological process will be, because the health, let's say, the health of those bacteria will be better the higher the number of PTMs. So yes, we know the proteins and we know the sites of modifications.
Thanks for that, Pedro. No, that's great. Oh, we have 2 more questions. Question for Pip. Would you like to field that one from Ashley Granter? Oh.
Unmuted. I'll just read it. Have you explored the benefits? So I'll just read it out. Have you explored the benefits of using soil fungi to improve soil condition and reduce reliance of phosphorus-based fertilizers? So in a short answer, I'm not sure. It might be something that Josh is doing, but I can certainly pass that on to that soil project. Jenny might be able to answer. She's going to come in.
Not at the moment. I think we'll take it back to Josh, and if he's got a comment, then we can contact you directly. Cool. James, there's a question for you from Godfrey.
Yeah, so this is a good question. I guess there are a couple of different ways that we can approach this. One is working out how we change those microbial communities. So microbial community structure seems to be quite reproducible if you can keep the conditions the same, and that's obviously a big challenge in engineering. So some of this is being able to demonstrate that the community changes and then point out where the variability is within the process. And then the other is to start directly manipulating how the community behaves, for example, by giving substrates. Again, actually what Andrew was talking about, potentially you can use other substrates to immobilize the microbes that you're particularly interested in, and retain them within the community in a more efficient way. So and of course, what you feed them, you know, it comes down to basically you are what you eat. So if you can control better the process of feedstock composition, you can also improve those processes. And actually, well, there's lots. I could go on for a very long time, probably.
Thanks for that, James. Do we have any more questions from our community, from our participants? Nothing at the moment. I've actually got a question, and it's pretty much to all of you, actually. So we'll do it one at a time. But the question is, thinking about your technologies, your science, and its role within the water system, what is the biggest challenge that you face taking your current science and technology forward? And I'm probably, we'll go sequentially. We'll start with Jai.
Thanks, Jenny. I mean, I think with electrochemical technologies for water treatment, they need to be specifically targeted to the particular application. So you need to know what's roughly likely to be in the water that you want to remove. So whether it's heavy metals, or whether it's organics, or whether it's this mixture, that's probably the biggest challenge, is understanding the composition. Taking it forward and implementing it then requires compliance with all sorts of regulations. So for example, on the Centrica Storage system, taking it to the platform or taking it offshore then requires it taking it to the offshore platform versus a terminal onshore are two different requirements. Both have to have compliance with fire safety regulations. So it's the ATEX compliance. But then moving to the offshore means that you've got to make sure that it's sufficiently lightweight and versatile and resilient for that particular challenging and demanding environment. So actually, those are the putting it into practice, I think, is the or understanding the conditions of how it is to be used in practice, I think, are the hardest parts to learn about. Yeah.
No, I think there are many challenges in translating research into practice, but that was really interesting. Thank you, Pedro. You're really at the start of your journey with this technology. So, you know, what are the next challenges that you're going to address?
Thank you, Jenny. We are, but having said that, the technology has been, the proteomics and mass spectrometry has been around for decades probably, and certainly the technology that we have used is widely available. I think the challenge for us now is identifying industrial partners really, and if industry is interested in taking these forward. So if you're interested then in exploring PTMs in wastewater treatment, then get in touch and we'll give it a go. We are ready.
Thanks. Great plug. James.
So challenges for us, obviously the microbial communities are massively complex, and for me, there is, like I say, the second project that we were involved in, actually understanding what changes at scale as a biologist is a big challenge. Maybe as a chemical engineer, it's less of a big challenge. But there are certain things that we can't easily test without going to large scales. Things like gas solubility are very different in a vessel that's 22 meters tall compared to one that's only half a meter tall, just because of pressure differences. So that's a really big challenge. And I think within sort of domestic wastewater, one of the
No, thanks for that. I want to give Andrew and Pip a chance to answer the question about challenges, and then we actually have another live question for one of our participants. So Andrew, where do you see the biggest challenge in moving forward?
I think for me, to echo some of the comments from James, everything's to do with scale. What are you trying to do? Because if you're trying to apply new technology to wastewater treatment plants with these billions and billions of litres of water which are being processed, they're very different than if you, for example, you are trying to make some sort of small plant water purification to attach onto a hospital to try and, you know, clean up what is normally very heavily pharmaceutical laden wastewater, or a care home even, things like that. Or it could be you're using something for water treatment, you know, in a fish farm, you know, and is that a sort of a landlocked type fish farm, or one which is part of, you know, sort of a penned fish sort of a zone, you know, sort of in a lock on the, you know, which is part of the sea. So from my point of view, the thing which I'm addressing next, haven't talked about it, are the possibilities of taking the material and actually going to the next technology level in terms of some sort of device or some sort of construct. Is it going to be filtration system? Is it going to be taking these particles and making them into the equivalent of granulated activated charcoal, immobilizing them in some way, so that you can then have a better idea as to what type of sort of like problem that you might be able to address with them. And I think until that's been done, you know, I don't think it's, you know, it's we're not going to be starting to say, hey, Yorkshire Water, you know, will you suddenly like to sort of actually sort of, you know, take some of these materials? Of course, one of the things that James did, he obviously mentioned some of the issues about cost with them and their processes working on. There's also the regulatory issue in, you know, you can't just do experiments easily on that sort of scale. So having those repurposed sites would actually be, you know, very useful in that regard, to take something from a lower technology level up to prove that it's actually effective at the sort of scales that you might come across in real life.
Thank you, Andrew. That's yeah, it is a challenge. Okay. And Pip, finally, just a comment on the challenges for sustainable drainage systems.
So again, probably similar to what Pedro said, less about the science for us, because we're in the business of knowledge exchange. It's more the uptake. It's kind of starting to stimulate the demand and the understanding and the education within businesses of the opportunities that there are for sustainable drainage, and making sure that that is considered early on in the development or the retrofitting of schemes, and really promoting awareness of the opportunities for sustainable drainage and the many applications for it. So that's where we come in, as I say, less the science, more the kind of putting that message out there that there's a real opportunity for small businesses to use this as a growth opportunity.
Thanks for that, Pip. We go back to the Q&A tab. Rambir is asking which of the sustainable development goals do you see synergies and trade-offs with, and what policies, directives will be adhered to and work towards post-Brexit? I wonder if anybody would like to tackle that one from a perspective of your process, and perhaps even Jill might be able to step in from Catch's perspective and think about the role of Catch in supporting businesses with their post-Brexit.
Hello, yeah, not the old B word, the dreaded B word. Yeah, so we've been working with our members and associates, and also our other regional cluster groups, taking the concerns from industry up to government. So through the Chemical Industries Association, working with them, you know, through all the Brexit negotiations. Any regulations around REACH and how they will affect the industry. So yeah, if there's any specifics people have got concerns about, then yeah, please get in contact with me and let me know, and I will make sure they are raised.
Okay, that's great. Thank you, Jill. Is anyone feel able to comment on sustainable development goals with regards to wastewater treatment? Is it something anyone feels?
I'm happy to comment on that. I think you're right. I think that's the context and the framework that we need to take into account, and it is a very, very useful framework. I think that most of what we have spoken about today is perhaps directly or indirectly relevant to some of the Sustainable Development Goals, to be honest, including clean water and sanitation. We also have life below water, and that's where the pollutants, so Andrew's work with capturing pollutants is coming in, for example. There is also an SDG related to climate action, and I think Pix was commenting on climate change and how that is going to change the context really of water treatment. And overall on the SDG number 3, which is good health and well-being. So I think what we have discussed today is relevant to the SDGs, and you're right, we haven't directly, I think, refer to the SDGs, but they are the as I say, the context that I think is underpinning a lot of our work.
And just, I suppose, from our perspective, the trade-off, I suppose, is at the moment these technologies are very much maybe not anaerobic digestion, but the refinement of that process is very much in development. And I suppose it's having the context of making those technologies ethical and affordable for a large proportion of the globe in order to meet sustainable development goals. It needs to be affordable, and it needs to be available and accessible from a wide proportion of the global population. So, and that's something to take into consideration through commercialization, I presume, as we move forward. Sorry, James, Karen.
Yeah, so I think, the whole idea that with SDGs as well is around circular economy and reuse and things like that. And, even the water companies are realizing this sludge isn't called sludge anymore, it's in bioresources, right? And so understanding that there are a lot of resources that can potentially be recovered. Biogas is the beginning, but actually, you can see down the way, if we can properly manipulate microbial communities in AD, we could recover much higher value products from those kinds of things. And that feeds into, a lot more sustainability, which is clearly important.
Oh, thanks for that. I'm afraid we've come to the end of our slot. In fact, we are a couple of minutes over. So just want to take this opportunity to thank everybody, all the participants for joining us, and all of our talks, speakers for giving such interesting talks today. As chair, I just want to share with you some of the contact details for the main contacts for this project. There's myself, there's Jill Mooney, oh crikey, sorry. Jill Mooney from Catch Technical Services, Alice North, who is from Biovale. So thank you everybody for joining us today, and a recording of this presentation will be available, and I believe that there will be a questionnaire sent out for feedback shortly after this meeting. So without further ado, I'd like to say thank you and goodbye.
Thank you.
Thanks, Jenny. Thanks. Bye.